All right, friends. You should have, by this point, been through podcasts dealing with lots of chemistry stuff, but ultimately carbohydrates, lipids, and proteins. This podcast is focusing in on a particularly important group of proteins called enzymes. Without enzymes, nothing happens in biology, or almost nothing happens in biology. All the chemical processes that occur in your body occur because enzymes facilitate them happening. So they're incredibly important. We're going to talk about everything from DNA replication, DNA translation, transcription, cellular respiration, photosynthesis, these absolutely quintessential processes of life, processes without which life as we know it really wouldn't happen, require enzymes to make them happen. So enzymes are really one of the fundamental aspects of biology that we need to understand if we're going to understand how life works. So I'm going to do a brief basic info about enzymes, stuff that's probably very similar to what you might have learned in honors biology or biology 1 some years ago. And then we'll go into a little bit more depth. All right. Enzymes are biological catalysts, okay? There are lots of catalysts in the world, okay? Whoops, my board's the wrong way, pardon. There are lots of catalysts in the world. They do not have to be anything that is biological. They can be inorganic substances sometimes. But in biology, we rely on enzymes. They are biological catalysts. What's a catalyst? A catalyst is anything that speeds up a chemical reaction, okay? Enzymes are a particular type of protein that speeds up chemical reactions, particularly biological chemical reactions, okay? Now, usually enzymes, as I said, are protein. Sometimes they also can be made of RNA. It has come to light in recent years that RNA is not just a form of genetic information type material, but it can actually do things. So that's a really amazing piece of information and very important to understanding how living things work. We are going to focus on as proteins as enzymes, understanding that as we learn more about biology, we are learning that other molecules can act as enzymes, have enzyme action as well. Okay? Now, they facilitate chemical reactions. They help them happen. Please understand, this means that they can help a chemical reaction happen, but they are not being used, like consumed in the chemical reaction. Like when we talk about cellular respiration, glucose plus oxygen is going to give us energy and the CO2 and water, okay? This is consumed, all right? But enzymes are not used up. They are more like, hold it right there. That, like a key, is used to open a door. We open the door. We do the chemical reaction, so to speak. But the key is not used up. I can use it again and again and again and again. Think of an enzyme as being like a key, and we'll go back to that analogy often, okay? Now, enzymes are said to reduce activation energy. What this means is if I have a boulder I am trying to push off a hilltop, that boulder, if I can just get it off the edge of the hill, off the edge of the cliff, it's going to go down. It's going to go down to the bottom. And it's going to go all the way down. And I'm going to have to do very little work. But I've got to push that thing off that cliff to get it down to the bottom of the cliff. Well, my push is activation energy. And what enzymes do is they lower the amount of energy it takes to get that chemical reaction started. They lower the amount of energy it takes to shove that boulder off the edge of the cliff so that it will go on down to the bottom where I want it to go. Okay? So activation energy is the energy it takes to get a chemical reaction going. And if I can lower the activation energy, I don't have to put so much energy into getting that chemical reaction going. Okay? We're not going to worry about that now. We'll hear more about that later on. We'll learn about this graph later on as well. All right. Enzymes are required for most chemical reactions in biology. You are a great big sack of chemicals, a miraculous sack of chemicals, I will grant you, but you are a sack of chemicals and chemical reactions. Most of them would not occur without enzymes helping them to occur, without enzymes lowering the activation energy, the energy that's needed to make that reaction go and do what it's supposed to do. Enzymes are highly specific. Again, let's remember the key analogy. This key is the key to my classroom, and it is highly specific to my classroom door. It will not open a single other door in the building, almost. Okay? So, it is very specific to its job. Enzymes are very specific too. Much the way this key is, they are fitted to particular chemicals and to helping particular chemicals engage in chemical reactions. Thus, since there are thousands of different chemical reactions in your body, there are thousands of different enzymes directly specified to make each of these different kinds of chemical reactions work. This last one, they really do control the processes of life. Enzymes control when chemical reactions happen, when they don't. If an enzyme is present, a chemical reaction will occur. If it's not present, it won't. By controlling what enzymes are where in your body at what times, cells can control their activities. It's really, really fundamentally important to how life works. All right, some certain terms that go with enzymes. First of all, let me set you up here with this drawing, and then we'll go look at the actual terms. This purple thing is representing an enzyme. This tan, yellow, whatever you want to call that thing, is representing what we call a substrate. A substrate is nothing more than a substance that we want to we want to cause to enter into a chemical reaction. Maybe this is glucose. I don't know. It looks like more like a disaccharide probably, and we want to break it down. We want to break that bond, let's say. All right. So this is the substrate we want the enzyme to act upon, and this is the enzyme. Now, you should notice that there is a peculiar shape in this enzyme. This enzyme has a place in it that actually is specifically designed to fit this substrate. And this place where the enzyme fits is called the enzyme's active site.
Am I on television?
No. Go away.
Backing up. Whoosh.
Now, the active site is where the substrate fits into the enzyme and where the enzyme can act upon it. Now, this, again, is very much analogous to the key. My key has to have a specific shape to fit the lock and make it turn. My enzyme has to have a specific shape to fit the substrate it is supposed to work with to do the reaction, to make the substrate enter into the chemical reaction we want it to do.
What's an enzyme? I'll shut up.
And once the substrate fits into the active site, then the enzyme helps. In this case, we want to break this apart So the enzyme, imagine putting a stick across your knee and stressing it in hopes of making it break. Well, that's essentially what an enzyme is doing. It will stress chemical bonds and make it easier for them to break in this case. But it can also force things, different enzymes that want to create bigger molecules can push them together and force them to come together. Okay? So once the chemical reaction's done, the products leave the enzyme, and the enzyme is left unchanged, looking exactly as it did when it started, ready to do the job again. So enzymes can be used over and over. Okay? So the substrate, here's your vocabulary: reactant which binds to the enzyme. This thing here is the enzyme substrate complex. That is a temporary thing. As soon as the enzyme has done what it needs to do to the substrate, they come apart. The product is what we get, the end result of the reaction. And the active site, already we've said, is the catalytic site, the place where the substrate fits into the enzyme. Okay? All right. Properties of enzymes. They are specific to particular chemical reaction. Just like the key to the lock. They are specific to certain substrates. Essentially, what holds a substrate in place in an active site, remember, there are many types of chemical bonds. We have talked about covalent bonds, we've talked about ionic bonds, we've talked about hydrogen bonds. But I don't want my key to fit permanently into my lock. I do not want to stick my key in and have it stay in place. Okay? So we want weak bonds that are strong enough to hold the enzyme and the substrate together until the job is done, and then that can be broken easily. Hydrogen and bonds and ionic bonds fit that bill much better than covalent bonds do. So you're not going to have covalent bonds hooking together a substrate and an enzyme. You're going to have weak bonds doing that. Okay? They are not consumed in the reaction. The enzymes can be used thousands of times in a row, many times per second. It can be amazingly fast. And they are unaffected by the reaction. Now, enzymes can, however, be affected by the conditions that surround them, by the conditions in the cell. Anything that can affect the structure of a protein, because remember, this is a protein, can affect the structure of the enzyme. Heat, we know, denatures proteins. It can make them change their shape. As heat is applied, imagine my key. If heat is applied to this key, if I took a blowtorch and applied it to my key,
I would like to see you do that.
I am going to end up heating and causing the molecules in the key to move very fast, and they will alter their shape. That substance will alter its shape because the molecules move so fast. That happens with enzymes as well. If you heat them, the molecules move faster, they alter their shape. And so the active site will not be shaped correctly, and it won't work the way it's supposed to anymore. In addition to temperature, things like pH can affect enzymes and their structure. Salinity, the amount of salt, can affect enzymes and their structure. All kinds of stuff in the solution, same solution as the enzyme, can affect them. Okay? Now, there are certain ways that enzymes are named. Most enzymes have ASE as the suffix of their name. Don't confuse that with OSE, which we have seen is common in carbohydrates as an ending. That's not it. ACE indicates enzymes. Now, oftentimes you can get a clue about what the enzyme does if you look at its name. Sucrase breaks down sucrose, breaks down that disaccharide. Proteases break down proteins. Lipases break down lipids. DNA polymerase, DNA polymer, is an enzyme that builds DNA molecules, and we're going to talk a lot about that later. Now, sadly, we also have some enzymes that end in IN because that's just how life is. Pepsin is an enzyme that breaks down proteins. Okay, polypeptides, pepsin breaks down. But most of the time, the enzyme names end in ACE. Okay, this is just an old idea, but I rely on it all the time because I use this key example all the time to talk about enzymes. The lock and key model is a kind of overly simplistic model of enzyme action where the substrate fits the enzyme like a key fits a lock. It kind of implies rigidity. The hydrogen bonds form between this little substrate as it fits into that active site. But what is considered to be most true now is this idea of induced fit, where it's not just a rigid lock and key, it's more like a couple of, really, Grace? It's a couple of like hands clasping. The enzyme has the active site, the substrate fits, but as you can see, the enzyme kind of closes in around that substrate to fit it more closely. So we have that 3D structure where the enzyme really fits the substrate, and there's a slight shape change of the enzyme to really closely fit that substrate. This type of shape changing is very common in proteins. We'll see it a lot because proteins do work. They do all the work. And things that do work change their shape. You do work all the time. You wouldn't do a lot of work if you were rigid, if you were stick straight. You do work because you can move around. Conformational changes, shape changes, are what allow proteins to do work. Conformational just means shape change. Okay, and in this case, it's like a hand fitting a glove. Okay, and what that does is it really does bring the chemical groups that are supposed to react together into positions where they can interact. Okay, all right. Now, okay. Now there are chemical reactions. We've already learned there are chemical reactions that build big molecules, like a big molecule of starch gets built from a bunch of monosaccharides. But there are also chemical reactions where we break down big molecules into smaller ones. Enzymes catalyze both those reactions, okay? Synthesis. Synthesis is where we build things. So in the case of synthesis, an enzyme is going to use its active site to bring molecules in together, to take a small molecule and a small molecule, bring them together to make a bigger molecule. A monosaccharide and a monosaccharide, we're going to bring them together and make a disaccharide, okay? But in digestion or reactions where we break down, digestion is kind of, we think of digestion as happening in your stomach, but digestion is an overarching term to talk about breaking things down. When we digest a large molecule, the active site is going to bind to the substrate, and like I said, like if you have a stick and you bend it across your knee, you're stressing chemical bonds. It seems to act mostly like that, making it easier to separate those molecules. Okay? Now, that's it for this introduction to enzymes. We're going to talk about factors that can affect enzyme function in the next podcast. All right? So, thanks for watching. I will catch you in the next podcast.